This is a neuromuscular junction with presynaptic membrane, postsynaptic membrane, and synaptic cleft. The postsynaptic membrane is on the junctional fold of the sarcolemma. When action potential reaches axon terminal, it releases acetylcholine through exocytosis. Acetylcholine released binds to the nicotinic receptors on the sarcolemma and increases its sodium-potassium permeability. More amounts of cations, that is sodium ions, enter the sarcoplasm and increase the potential inside the cell. There is a slow increase in the membrane potential until it reaches a threshold value. Then there is a rapid increase called depolarization. Action potential is produced which spreads to the T tubules. The membrane of the T tubule has a receptor known as dihydropyridine receptor, also known as long-type calcium ion channels. There is a sarcotubular system in which a sarcoplasmic reticulum cistern is present, which has a membrane receptor known as ryanodine receptor. The cistern is rich in calcium ions. When the action potential reaches T tubule, the DHP receptors detects the voltage change and changes the ryanodine receptor which it's physically linked to. Because of this, calcium ions leak into these sarcoplasm from the cisterns. The calcium ions in sarcoplasm now bind to the troponin C. So as we can see, they create some conformational changes in tropomyosin so that it uncovers the myosin binding site on actin. This we'll see later leads to formation of the cross bridge. Myosin head has two sites, ATP binding site and actin binding site. The ATP attached is hydrolyzed to ADP and inorganic phosphate by ATPase. Myosin head gets energized and binds to actin. This is the power stroke, in which myosin head flexes and forces the thin filaments together. The detachment of myosin head from actin is by binding of another ATP, and when this ATP is hydrolyzed, the cross-bridge cycle continues. Relaxation of the muscle is by active pumping of calcium ions back to the sarcoplasmic reticulum by the sarcoendoplasmic reticulum calcium ATPase. This decreases the sarcoplasmic concentration of calcium. Subsequently, it is removed from the troponin C and hence the myosin binding site of actin is covered again. This leads to cessation of the cross-bridge cycle and relaxation of the muscle.